Much, madam, and good morning to all of you. So, very good morning to all of you. So, today I'm just planning to talk about some recent as well as some advanced concepts in plant breeding and genetics, and most of the things you won't see it in your book chapters. So my idea is to not to go in depth about this topic. Just I wanted to give you, glimpses how practical breeding works across globe, especially in international research organization as well as in multinational commercial seed companies. Okay, and it three actually. Basically, this is how actually plant. So before going to the details of the concepts, my sincere acknowledgement to this to the Bill and Melinda Gates Foundation. Most of the works have been carried out with the generous funding from Bill and Melinda Gates Foundation, as well as from my year we got more than 50 crores for implementing such kind of advances in breeding, as well as a lot of other collaborators associated in this project. So this you may be knowing that that now we have been attained highly self-sufficient in food grain production. Right, more than 300 plus tons have been produced in the last few years. Every year, in fact, in the cereal crop, in the staple crops. Whereas the current population, that it's around 143 million, 1.43 billion. The current, and it is going to be projected around 160 or 1.6 to 1.9 billion in the next 20 years. So how to feed them? That is the idea, right? And we are all, in fact, the sole responsible guys to give high yielding varieties to the farmers as well as to feed the general public, isn't it? So the idea is how to meet this this gap. One side, this population is going up. The other side is we are under pressure to produce high yielding varieties. So there are certain concepts. That is what I'm planning to highlight those concepts. So these are all a lot of some of the breakthrough technologies in genetics and breeding in the last 100 years. It's not a very exhaustive list, but it's kind of a very I'm just highlighting that starting from your Mendel principles, then nitrosis, then this is the green revolution era. Then there has been mutagenesis, mutation breeding. Then in the last 20, 30 years, we have been working on molecular markers, QTL mapping, association mapping. These are all kind of. Then there was NGS, next generation sequencing, 20 years back. Then now we are moving into the genome-based selection. It's not one or two genes are controlling the traits. The whole genome is contributing for the trait improvement. So we are moving towards that those kind of concepts. So there are a lot of strategies are there to increase the yield. This idea is how is all about how to increase the or how to release the varieties rapidly, right? So before that, I wanted to introduce a couple of words. Anybody knows what is the meaning of this genetic gain? Anybody? Have you come across these words? Genetic gain? Yes? Somebody say yes. Okay, good. So usually the yield gain is that what is the total production from the variety? Okay, from the breeding by releasing a variety, you can just measure the value of that variety. That is called yield gain. Actually, that also comes from different practices, not only from genetics. When you apply some fertilizer, when you go for some high end package of practices, you will get the yield gain. Whereas genetic gain is the kind of improvement that you made it only through your genetics and breeding concepts. Okay, you cannot put the responsibility to the others. Whatever that we are doing in genetics and breeding, right, for the improvement of the genotype, that is called genetic gain. Okay. So this is one of the kind of classical examples where this genetic gain been realized in animal breeding program. In 60, 70 years back, the average weight of the chicken is just 1 kg. Now in the animal breeding program, they could get more than 4 kg per genotype. So which means that they could 7.5% of genetic gain per year. So there was the base for such kind of it's only one gene, right, which is called a dwarfing gene. Okay, and it has been associated with so many positive traits. Okay, like nitrogen response charging, shorter type. Okay, so these one or two genes provide a tremendous, gain in terms of, the visual, measurable traits, characters. Okay, so this is called, genetic gain. Uh, but after 30, 40 years, after this green revolution, we don't find much genetic gain in any breeding program because we don't have any technologies. For example, this fan is invented 100 years back. This is exactly what the technology was there and been now also. Okay, there is no change in technology, right? So the same way that 60, 70 years back, now we found some kind of technologies in breeding, right? Works really very well. But last 50, 60 years, we don't have that much, kind of revolutionary technology in breeding. So that the output from the breeding program also very slow, right? So this is, for example, this is the overall genetic gain that we could get from different breeding program, international breeding programs. For example, okay, genetic gain. I actually I need a lot of time to explain all those things. Being a student, it's really difficult for me to explain. So, genetic gain, you can measure the genetic gain simply. For example, you take the varieties that are released in different years, 1960s, 70s, 80s, 90s, 2000, 2010, 2020. Okay, so there are varieties released in 60s, there are varieties released in 70s. Okay, so let us take all the varieties, then we will conduct one trial now. Okay, you take 60, 70 varieties are released in rice for the last 60, 70 years. You conduct a trial here, simple RBD, RCBD trial, right? Then we can measure how much genetic gain has been attained in the breeding program. Okay, then by comparing the varieties released in this last 10 years versus the varieties released in the last 50, 60 years. So that kind of trial will give you an idea about how much genetic gain has been obtained through the breeding or genetics means or concepts. Okay, so that is how we have to measure the genetic gain. Whether our genetic concepts like the dwarfing gene has worked really well or any other technology through breeding or your QTL mapping or marker step breeding, right? Or any other your generation mean analysis, whatever the concept that you have been using actually for developing varieties really worked well. Is there any yield advantage of the varieties released in 60s, 70s versus the yield advantage of the varieties released in the last 10 years? So we can measure the genetic gain. Okay, the difference between these two values will give you the value of the genetic gain, right? What I'm saying that process has been become so slow. We couldn't get tremendous improvement in terms of yield by applying the kind of concept that you have been studying in your textbook. It has become obsolete. Okay, we have been following a lot of conventional approaches which is not giving any yield improvement that we want to see, right, from the field or from the varieties that we release. Okay, so there is a kind of it has been stalled in fact. Okay, we couldn't get any tremendous progress in yield improvement by applying our genetics and breeding concepts. Okay, that is the statement I want to give you, right? And it is not the case only in isolated breeding programs all over world. The genetic gain, the improvement in yield, okay, by applying this genetics and breeding concepts has been slow in the last 30, 40 years. So that is what it's showing. It's less than 1% actually. And it's going to be very slow in the next 20, 30 years. Also, if we keep on following the concepts and the methods what has been given in your plant breeding textbooks, it's not going to be useful. Okay, it's all obsolete, right? We can produce varieties, right? We can release varieties, but it's not going to be the amount of no time we are wasting in the field or in the lab not going to be translated into the yield improvement. Okay, so how to improve it, that is a question actually, right? This is the formula, right? Unfortunately, most of our breeding books they don't talk about this kind of formula, but it's really a very very important, the mother of all formula that you have been studying in plant breeding and genetics book, right? So this is called genetic gain, the formula to get high yield, right? So this is selection intensity. I stands for H stands for no accuracy or heritability, and this is no the additive genetic variance, right? All those things to be improved in a shorter period of time. That is how you can maximize the genetic gain. Now you understand the difference between yield gain and genetic gain. When you say that we have got yield gain, that also comes from the agronomic practices or by controlling pests and diseases. Right. Many other disciplines also contribute for yield improvement. Whereas genetic gain, right, whenever you use the word genetic gain is a very, very specialized formula, right? You have to use these no factors. These factors, in fact, are contributing for genetic gain, right? So this can only be done by the breeders because you do have a control over the selection intensity and the heritability, and you can control over the variants that you are generating from the breeding population, and all those things can be obtained in a very time. That's how you can release varieties rapidly year after year with very with maximum advantage, yield advantage. So again, it's a very practical way how we do the breeding programs in international breeding programs or very successful breeding programs or multinational seed companies breeding programs. In the left side, if you see uh, or in rice or self-pollinated species or or cross-pollinated species, there is a line development program. You have to develop first inbreds. Okay, then the inbreds can be released as a variety by testing different location if it is a self-pollinated species like rice and wheat or pulses, or we have to make some kind of crosses like in pole millet and hybrid, and it has to be tested all over locations for release. For any breeding program, we need to develop some successful inbred lines. Okay, that is the first step of any breeding program. Usually in conventional breeding programs, we follow this kind of approach. We make some random crosses, or with the kind of conventional wisdom, these breeders, right? They do select some kind of parental material. They do cross, and we will go for F2, F3 to F7, right? And yeah, either it can be released as it will go for MLT multi-location trials for testing further, or we can make hybrids. On the other hand, so this is a kind of new concept that we are implementing on a pilot scale in different crops. This is called a rapid breeding cycles. So we have to select the parental lines very wisely, right? Then instead of F7, F8, you can select the better lines in F3, F4 itself by using the concept of genomic selection, right? I'll come to that point later. Then you can also improve the accuracy, which means the heritability, by following some very advanced field trials like a sparse test, which means that you don't have to conduct all you don't have to spread all genotypes or lines across locations. You can select some subset of the lines in different locations, but by putting some kind of common checks and testers across locations. So don't expect me to explain everything because of the time, but my idea note down these keywords like a sparse design, right? It's all no, it's all totally different from your RCBD, SPARSC, sparse design, right? Or partial replications. Go and Google it. Google is a god nowadays. Okay, then you will come to know that how this approaches was. So we can improve the heritability that way. And as I said, this time. This can be done quickly by selecting lines in F3, F4 itself rather than F7, F5. And there are other complementary approaches are there to run this breeding pipeline. Like we can also go for genomic selection. Then there are like speed breeding approaches are there, right? One of the structure is already established in a parry breeding station also, and we can also digitalize the breeding program. So the important point as a breeder, I wanted to emphasize that the best parents produce best. Okay, best parents produce best progenies. Um, that concept is called genomic selection, right? You might have heard the word QTL mapping, association mapping, right? Especially for the quantitative traits, these kind of approaches are obsolete. The so-called QTL mapping through biparental crosses or association mapping using LD-based concepts are obsolete, not working for quantitative traits. I don't find any varietal improvement program from association mapping or QTL mapping because they cannot give you 100%, they can't capture 100% of your genetic variants. You will get one or two QTLs for yield, right? When you transfer that QTL through your marker step breeding program, you can capture only 10 to 20% of the total variability of the trait. It won't work. It never was, right? It will work for traits like disease resistant, right, or some qualitative traits, mostly controlled by qualitative way, right? Whereas polygenes for yield, grain yield, right? This QTL mapping and association mapping don't work, and we don't see practically any no, the applied breeding program uses the output from QTL mapping and association mapping, whereas the genomic selection is no kind of a very contemporary approach where it uses the whole set of our genome or the genome of the crop, right? Because these quantitative traits are not controlled by few genes. There are so many genes, there are a lot of interactions are there among them, right? So many pathways are working on trait improve on the final trait makeup. Okay, so there is a concept called genomic selection, right? This genomic selection we can use it for selecting the parental lines for the crossing program, because by this approach we can measure the breeding value of the parental line. Okay, so breeding value is a kind of a value assigned to a particular genotype, right? You have hundreds of thousands of genotype. When you go to paddy breeding station or millet breeding station, there are a lot of accessions inbreds, right, or lines evaluated every year. By using this approach, we can give a value to that particular line that is called breeding value. Like we can give some breeding value for humans also, right? Everybody has some breeding value, right? If you do this genomic selection approach, the genomic selection approach you can go for you have to do this complete comprehensive phenotyping of the genotypes, then simultaneously you have to do some genotyping also. So by using some statistics genomic selection model, you can measure the breeding value of the genotypes, right? So this will be the starting point for your pollination program, rather than taking some individuals based on our conventional wisdom. Conventional wisdom means somebody told no, or your previous breeder, or you do see yourself from no field testing. Okay, these genotypes are good. Visually, you have taken some data, right? Then you use them for no breeding program, rather than you can just go for no kind of very educated or no very systematic way of selecting the parental lines for your breeding program. Okay, so that, as I said, that best parents produce best progenies, right? If you think these concepts very deeply, we are all of the genomic selection in humans. In animals, it works really well, actually, right? As I said, in that chicken breeding program, or any animal breeding programs all over the world, this genomic selection concept really works well. And we are all, in fact, all humans that are we are sitting here right now. We are all the products of the genomic selection. Our forefathers are carefully selected. You can go back several generations, right? Then we are the product of the genomic selection. Best parents produce best progenies, right? So that is how this concepts work. So you can select that, parental lines for your pollination program. Then you can develop the population. The F3 F4 population. In F3 F4 population also you can apply the genome model. Then you can select the best progenies, and that progenies will be used for line development program, or it can be again recycled, right, to generate new crosses. Right, because this way we can enrich the superior alleles. For example, the number of superior alleles or the favorable alleles in the population will be, some X number, right, X percentage. By recycling the progenies from the first round, like you make crosses, then you go for F2, F3, F4, maybe 2000 lines, you select some 5% of the best lines, right? Then again you can recycle that 5% to the next round of cycle, breeding cycle, so that you are keep on accumulating the superior alleles in the population. That is what we are right now. Okay, so we have been enriched with a lot of superior alleles for last thousands of years, right? For your physics or for your intelligence or for certain traits, right? We are all the product of genomic selection. We are enriching the superior alleles, right? That is how exactly works in this breeding model, right? So there are so much of thought process gone into, right, how to select those parental lines, how to know how much selection intensity should be applied in the population F3 F4, right? So that, and there are a lot of testing models, as I said, that partial reps, sparse design to improve the heritability. So all those things will work in this breeding pipeline, so that all these factors then you can apply in this formula, so that the genetic gain will be improved shortly, right? And if the genetic gain improves, their yield gain also will be improved, right? And if you apply, the time here is not only the early generation selection, also you can this kind of rapid generation advancement techniques like double haploids or speed breeding, right? Such kind of technique if you use, and you can produce new varieties just in 2 years. So 3 to 4 crops you can use using the speed breeding or rapid generation technologies, and this early generation selection combined with the genomic selection concepts, you can produce new varieties just in 2 years. That is the idea actually, and we are testing this kind of concepts in 4 or 5 crops. Well, you might be knowing that these are all some kind of high throughput technologies, genotyping technologies, right? You might have come across all those kind of tools and technologies. Apart from that, I'll come to that point, and I really don't know whether you might be knowing this kind of thing. Long back, how many of you are using SNP based selection here? Any students are using SNP based selection? Mostly we use SSRs, isn't it? So now the breeding pipelines are now moving towards SNP based trait selection actually. For example, you have a disease resistant for blast or shoot fly. Okay. So you have already identified this will work for no qualitative traits, right? So this is your rice field. You have put up so much of lines, 200, 300 MABC lines, Marcus, backcross lines, Marcus breeding backcross lines. So we collect the DNA, right? Then you run the SSRs. Then it takes 3 to 4 months time. So there are a lot of approaches are there, like CASP-based genotyping. So the QTLs, right? This CASP is SNP based approach. So in fact, before taking the nursery, you can isolate DNA from seed also, right? or if you want to take DNA from seedlings, you can take the DNA. You can just take a punch. No, you have a stapler, right? You go with a stapler. You just take one kind of strips of those seedlings. Then you can go for high throughput DNA isolation, and you have a cask marker. So then you will be able to identify which of those genotypes in the nursery, having right allele, right? So this whole process, you can do it in 5 days. So you don't have to. The point is that you don't have to do no selfing or crossing of hundreds of thousands of lines. You can just select only those interested lines. So this is a kind of no kind of things are going on. And as I said that these are again kind of new concept like a mid-density platform. So mid-density platform is either there are different types of platforms. So there are low density, medium density, high density. High density means that you scan entire genome, you will get millions of SNPs, right? Or you will go for one or two SNP based low density platforms. So this is another optimized number of SNPs, like 4,000 to 5,000 SNPs, and we also created such kind of pipelines. I mean SNP platforms in millets and pulses as well as in rice. So 4,000, 5,000 SNPs are no more than sufficient to do multiple genomic experiment like QTL mapping, association mapping, genomic selection, or background selection, right? So this kind of tools also we can generate. These are no kind of technologies. People will be using this kind of technologies for selection. Well, this is kind of a genotype. Oh, it's getting late. So there are a lot of genotyping, phenotyping options are coming up, including load cell based phenotyping to measure the water use efficiency, or drone based, or a lot of high throughput platforms are there for phenotyping your traits. And we are also digitalizing the whole field. Actually, we don't no longer taking the data from notebooks. So you can just use some kind of Android based applications. You can take the data. In fact, you can scan the plan. It will give some kind of measurement, like artificial intelligence based approaches, right? This is a speed breeding concept. I hope that you might be knowing that. So idea is that what we are trying to say that we are integrating all these concepts, new concepts in breeding, like genomic based selection to identify parental lines, right? Then you can use them, the best parental line, the best of the best parental lines in the crossing program or pollination program. Then you can use modern approaches to select the plants. Then you can recycle them. Then you can use digital and speed breeding approaches to reduce the times. All these things will in fact help you to identify the best of the best lines. That best lines will in fact associated with higher yield. So apart from that, there are a lot of approaches that you might be knowing that we are also doing for, especially for a qualitative trait or genes control. Easily you can find out those genes for iron and zinc. Then we are using a lot of whole genome transcriptome based approaches to identify the genes. Then for blast, we used whole genome transcriptome as well as validated those genes using association mapping. So once this is for drought. So once we know these SNPs, that can be used in the selection program. So thank you so much. I know that these are all the concepts are not mentioned for the MSc PhD students, but my idea is that maybe it's this kind of slides for kind of for meant for the breeders. Actually, we are working in the breeding program, but you guys are the future breeders, isn't it? Maybe working in a kind of public sector breeding program or commercial breeding programs. Probably you can note down some of the keywords from the slides, right? Then really it's going to be useful actually for your future. Thank you so much for this opportunity. You can ask any questions. I'm confident I can, whether I'm giving answer or not, but there is no stupid questions. I have a luxury to answer stupid answer. Okay, so ask questions, no problem. Yeah, actually, as I said that in all over world that genetic gain has been less than 1%, but they are trying to improve the genetic gain by at least 2%. For example, who are the breeders sitting here, we are using the just 5%, 10% over the check varieties for release, but for several years. But when you see the varieties released in 2000 and 2023, if you see the yield difference, maybe 10%. So that is the crux of the problem we are facing. But by just shortening that variety release, so I missed many things actually. There is a rapid varietal replacement program. So idea is we have to replace the varieties once in 3 years. Then that has to be kept like a check. So the 10% will be the 10% of that variety released. So we have to keep on changing the checks frequently. We will have a new target to replace so that it will keep on moving. By using these approaches, maybe in 2 to 3 years, we'll be able to replace the new varieties rapidly. That is a way. And in fact, most of the breeding programs now, MNC breeding program, I have gone through many of the breeding programs, as well as international breeding programs, not only the CG breeding programs, but other breeding programs. The selection is not based on only the visual traits or field phenotyping, what we are doing. They completely transformed into the SNP based selection, as well as genomic based selection. So for example, many breeding programs, if you see in India, they do only phenotyping, but or somewhere in USA, then they give the advice these plants, and they do select them and test them. They do only testing comprehensively. Most of the selections are totally into there are two things I've been also, yeah, I'm also listening this, and I'm also part of this, and 20 30 20 last 15 20 years we have been doing a lot of markers breeding program or QTL mapping program. We should be we should bond this accountability. Many of the QTL breeding program are done in the last 20 years only for publication. We have been done for I could see more than 1 lakh QTL mapping research articles, but how many of them are translated into that 1 lakh into variety? Not more than 30 40 numbers. I'm saying that, which means that just for sake of publication, we have been done so many things, and obviously anybody who wanted to give money is really felt bad about that. We ourselves actually underestimated the science and done something wrong. Nevertheless, the science is very robust. And in fact, we released when I was in IRI the first provitamin A hybrid in maize worldwide. That was the first release. But the donor line through Marcus breeding program, that donor line of a high provitamin A was identified by the Harvest Plus group at USA, and through these approaches only, association mapping approaches only. So which means that the concepts are very robust. And the only thing is we need to define the materials method in such a way that it will really target the real alleles. And in rice also is really working well, some of the disease resistant programs. Another thing is, of course, we cannot translate all breeding programs, like with the modern concept. Let us take any 1 or 2 breeding program, either rice or millets or somewhere here. Then when we demonstrate with, put all the resources there, and if you demonstrate the results, then of course we will be able to convince them. This is what I'm saying, that this is not for publication. We haven't published anything in the last 5 years. We spent nearly 50 crores. It's not for of this work is not to publish something like that. Of course, that is a spillover. Only thing is we wanted to demonstrate whether this kind of high-end rapid breeding pipelines works really well to capture higher alleles and superior alleles very rapidly. That is the idea. As I said that, see, when again I'm telling the same story, QTL mapping is just done by 100 genotype, 120 genotypes with 40, 50 SSRs. Then we publish it. At the end of the day, many of us are now doing QTL mapping with SSR markers. Of course, there are a lot of other limitations that are associated with that, but we should avoid that kind of thing. A person who evaluated, sitting in Delhi or some other place, he only see the output. He doesn't understand whether it should be done genome-wide SNPs. Like all association mapping, we should stop doing association mapping. It won't give you. The software will give some output, we will publish it, but it won't give you the real allele. That's why we spoil the program by ourselves. So at least I'm saying that at least 1 or 2 pilot programs we can take it, and of course, as you said, and I'm very positive about that, at least hundreds of theses can be created using this few slides what I mentioned. Actually, always we'll repeatedly doing the same thing, and See, ultimately, we have to do something for the society or the farmers' benefit, actually. And these people also can learn something more about how practical breeding works, actually. I really don't know. I'm really sometimes no felt bad. It's need to be no discussed very elaborately. In fact, no, yeah, probably. I wish that probably no professors, no all heads are sitting here, but you can include no revise the chapter in such a way that no including all modern concepts also, because whatever my personal opinion, in fact, what I've been teaching in breeding, 95% we are not doing in real time. Those concepts are obsolete. I really don't know why we are studying all those things actually. So when you revise the chapters, in fact, so it's all over India, not only to TNU. So we need to include some chapters. I mean, and which really give some practical implication to the breeding program actually. For example, we start with we studied the theory of inheritance, cutting the rat. I really don't know why we have to say some negative thoughts, isn't it? Then going into positive, the genetics, but if you say the prelude, not chapter one. You used to have a habit of reading books, the prelude of genetics book, right? It gives some kind of no useless concepts and ideas. I wish to after Mendel you can cut down several chapters, come into some kind of no as yeah, you need to understand the evolution of these markers. When we started this kind of marker-based approaches, first isozymes comes in 70s and 80s. Then there is a marker called RFLP. Then some markers called RAPD. These are no everybody known common markers actually. Then the SSR comes. We are using SSR because you are using a Nokia because you don't have a no Samsung. Would you like to use Samsung or Nokia now? Yeah, right. So that point of time, we didn't have a technology to use SNPs. SNP means single letter. You can scan whole genome, right? Every has maybe a probable polymorphism, isn't it? When compared to the second genotype, isn't it? Second sample. So now we have a no kind of Nokia or Samsung or no kind of Apple. Why we have to use Nokia 2210? You might not be knowing that we used such kind of phones, the small kind of phone. So that is the difference, right? So avoid using SSRs because probably no resources are not there, or we need to convert our whole breeding pipeline into